Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Emma Magavern. Emma is a medical doctor and a PhD candidate as well at the William Harvey Research Institute at Queen Mary University in London, working with Professor Mark Caulfield. And Emma, we were just chatting for a second before the show and mentioned that your first degree was in English. And you're actually not the first guest on this podcast to find their way to medicine and genetics via an English degree. The first one that I interviewed a couple months back was actually Nobel Prize winner Harold Varmus, who also did a degree in English literature. So you are in good company, not just on the podcast, but more generally. And it'd be interesting to start there, actually, because you don't get to meet very many medical doctors and geneticists that start in English in the United States where you come from. It's not even that easy to get into medical school. If you have an English degree, you probably have to do additional biology class and those sorts of things. So we're going to dive into that a little bit. You were also first author on a really great recent paper that looked at pharmacogenomics in a British South Asian population called East London Genes and Health. And I thought did some really interesting work looking at the relationship between genetics and statin use and development of cataracts, and also helped to shed some light on some pretty important broad medical questions. So without further ado, thank you for joining and welcome to the podcast.
Thank you. Thank you so much for the invitation. I'm very excited to be here and delighted to hear that you've been speaking with other English majors. That's news to me, but very welcome news. As you said, it's not the most usual path, and I do normally find that I'm in a bit of a club of one, having come from sort of the more humanities background. But I do think that the analytic approach that you get from studying humanities literature is broadly applicable to science as well. And when we talk about using genetic tools within sort of research to answer clinical medicine questions, you're talking about interfacing a lot of different disciplines. And I think that actually, oddly enough, humanities background gives you a pretty good preparation for doing that.
Yeah, that's true, actually. And the so many of the research questions and of course medical questions boil down to some combination of science, but also understanding humans in some fundamental way, right? Which literature can help us to do.
I hope so. I hope so. Strong proponent of the humanities, so preaching to the choir there.
Harold's PhD thesis had like a series of Beowulf quotes in one of the opening chapters. So when you're writing up, you can take whatever your favorite English literature is and try to weave that in there if you're feeling like it.
That's wonderful. That's a great story.
Yeah. So tell me about pharmacogenomics. What drew you to the field, and also you started as a medical doctor, but you decided to move into genetics, and you did I believe you did a course, master's degree in ethics as well along the way. So I'd love to just have a little bit of a picture of what got you to be interested in this field from where you started.
So it is a rather sort of convoluted journey, I must say. I really enjoyed my medical studies, but I always thought that the sort of scientific discovery aspect was very interesting as well. And I did do a master's in bioethics in Europe between my MD and then my postgraduate training in the United Kingdom. And I also was very fortunate to have the opportunity to do a master's in genetics with Health Education England, which is an amazing initiative that they make available to try to upskill clinicians so that you can get that clinical insight into using the genetic tools very translationally. which is what I've tried to do. So I think that pharmacogenomics is a really wonderful area because it ties together all the things I'm quite interested in and work on, which is using genetic tools for insight into sort of why different people respond differently to the same medicine. And there's a very clinical angle there, right? Because we prescribe lots of medicines as clinicians to patients every day. And, the way we live now, almost everyone will have medicine exposure in their life at some point now or later. So if you put together the pharmacology aspect, understanding how drugs work and how the body acts on the drugs and how the drugs act on the body, and then the clinical aspect of how we use those medicines in day-to-day clinical care, then I think there's a real opportunity to use genetic tools to get a deeper dive into some of the reasons why different people respond very differently to the same medicine. And that's pharmacogenomics in a nutshell.
So talk a little bit about East London Genes and Health. This is a really special program in the UK that I've talked about a couple times on this podcast, just because I think they've done an incredible job at powering some really novel research and also engaging really deeply with a community that's traditionally underrepresented research. So maybe you could talk a little bit about your work with that program, how it's different from some of the others that you may have worked on, and then talk a little bit about this research study that was recently published.
Sure. So Genes and Health recruits from a population in the UK, which are people who are Bangladeshi or Pakistani ancestry, self-declared. Now, that's really, really important for pharmacogenomics, genetics generally, pharmacogenomics specifically for two different reasons. One is South Asian communities generally are really proportionately underrepresented in genetic research. So when we talk about genetic research, of course, we know that different variants and different genes can be enhanced in extremely sort of diverse ways in different ancestry populations. And if we derive our conclusions from research, then that research maybe can't be generalized well across diverse ancestry, but if it's not representing those ancestries. So the issue is that about a quarter of the world's population now are South Asian ancestry, but the representation in genetic studies is very low. I believe less than 5%, but I would want to check myself on that number, but it's really disproportionately low. So it's really important to get that genetic representation from this cohort so that we're sure that we're reaching the right conclusions to translate to benefits for that population. The other thing is, if we talk about therapeutics, what we do in terms of licensure for safety and efficacy. So anytime we prescribe a medicine as doctors, we want to know that we're getting the best benefit for the least risk, right? Best benefit to risk profile. Now, the way that's established through a process, of course, clinical trials, and then the regulators evaluate that. However, clinical trials are a very special subpopulation, right? And from that, we're pulling this efficacy and safety information that we're then extrapolating for the global population. And we know that our ancestral diversity in terms of therapeutic population trials is not great, okay? So a lot of very commonly used medicine, I'll take for an example, a medicine called clopidogrel, an antiplatelet agent that's very commonly used after a heart attack, for example. If you go back to their licensure for safety and efficacy data after treating someone who's had an ischemic heart injury, some sort of a heart attack, there's no representation at all from these South Asian ancestry groups. So if you put together the error margin in that genetic data and the error margin in that clinical trial data, you can see that we really need diverse representation, particularly in pharmacogenomics.
And so what did when you move on to real world studies like the one that you did, what I think was very powerful about what you had is you have not just genetic information, but you have linked medical record data in the UK as well. So maybe you can talk a little bit about the scale of the data set and population, and then we can dive into this association that can you make statements about it being causal? I suppose you can tell me looking at the Mendelian randomization analysis, but maybe you could talk a little bit about the data set that went in and the hypothesis that you were going after.
Of course. So first of all, the East London Genes and Health is an amazing resource. So the PI is David Van Heel, and the team that's based at the Queen Mary University of London have done an amazing job. I think they've recruited about 55,000 participants at the moment. Now, thanks to their incredible work, the genetic data and the survey data that they get from participants at recruitment is linked, as you've said, with the health electronic record data. And that is things like hospital records and NHS digital, but it's also the GP medication data, which is really, really important because that's lifelong prescribing data. So that means we're able to get really good information about which medicines these participants have been prescribed by their GPs in a longitudinal fashion, and also how they've done in terms of health outcomes, as you've said, on a national scale, which is really valuable insight. Now, if you compare that to other really wonderful large-scale projects that we've all benefited from hugely scientifically, like the UK Biobank, for example, this is, of course, a much more diverse population, being all people of Bangladeshi or Pakistani ancestry, which is also a disproportionately deprived population. Well, of course, we know that with the UK Biobank, you're getting a very different group of participants.
Yep. And I want to just read directly from the paper here because I think you teed up the problem really well. And if I try to paraphrase, I'm just going to get points wrong. So you said in the paper, cataracts are a leading cause of blindness worldwide, particularly problematic in low and middle income countries with less access to surgical interventions. There's a reported association between statin use, which is used to lower cholesterol and blood pressure, and cataract risk. But this link is controversial and bidirectional. So some studies say statins increase cataract risk, and others, I guess, say it lowers risk. So it's messy. And there have been randomized control trials and meta-analyses. I'm actually paraphrasing now, despite me saying I was going to read directly. There are large observational studies as well as randomized control trials and meta-analyses that have found statins have a protective effect on cataracts, while others have found an increased risk. And so, there have been systematic reviews and meta-analysis that basically point to a potential effect, but a very slim one. What you did here that I thought was really interesting is you looked at this directly in this population, and you looked at people who carried or didn't carry the gene. You had people who were taking statins and those who were not. And maybe you could talk a little bit about what might be the heart of this conundrum where some studies see an effect, others don't, and some see it reversed.
Sure. So the issue with looking for sort of safety signals from medicines is that it's complicated, right? Different groups of people take medicines from the people who don't take medicines. And if you wrap up sort of how prevalent the medical conditions which predispose to another condition, which also sort of indications for medicine use in one population versus the other, it all is very mixed up and confused, right? So what we wanted to do with this was sort of a proof of concept study, as you say, using genetic tools to answer a clinical question and get a bit more insight. So what we did was we said, okay, there is this gene that encodes a transport protein. And we know from lots of other studies in the past that that transport protein is responsible for the hepatic uptake and then the clearance of statins to different degrees in different statins, but very high degrees in the most commonly used one. So if people have a relatively common missense variant in this gene, it means that that protein doesn't work as well, and therefore these people have exposures to much higher levels of that medicine active agent, okay? So we can say, all right, we've got a genetic proxy for people getting a much higher dose of this medicine. And can we use that to say, is there a relationship between the genetically predicted exposure to this medicine and the occurrence that we're interested in, cataracts, in people taking statins and not taking statins? So as you exactly said, we wanted to use genetics not sort of as a sort of research question in itself, but as a tool to answer this clinical question, which otherwise can get quite confused. I think Mendelian randomization, of course, has become incredibly popular in the past few years. And I think the reason that's incredibly popular is because it does the same thing, right? It says here's a genetic proxy tool that can untangle these complex questions, which otherwise are very much muddled up.
And maybe you can break down what you found, because you found that some carriers of the carriers of the gene are protected from cataracts, right? And then what does that tell us about the whole relationship between, or lack of relationship between, statin use and cataracts potentially?
So the thing I would say first starting off is that statins, probably clinicians, non-clinicians, everyone's familiar with this, but never hurts to remind people, statins are incredibly commonly used. So they're used, yes, for lipid disorders, but also for primary and secondary prevention of cardiovascular disease, which as we know is unbelievably prevalent now. So lots and lots and lots of people take statins, and we know that the benefits are very high. But because such a large swath of the population take it, there's very reasonably a lot of concerns about any potential side effects. So I think there was a lot of concern from people about this hypothesized relationship with cataracts. And of course, we're very fortunate in the United Kingdom to have access to surgery for cataracts, but not everyone does. So framing that. Now, in terms of what we found, as you so nicely said, there were studies in the past that said that they thought that statins were increasing cataracts or decreasing cataracts. But it's not that straightforward, because we also know that if you have cardiometabolic disease, which is a reason you would take a statin, you have a much higher risk of having cataracts and having young onset cataracts. So different studies have said that, for example, diabetes gives you between a 4 and 5 times higher chance of having a cataract, right? So that's just an example. So what we found was, first, if you do just divide the cataracts, young onset cataracts and cataracts generally, into sort of how common are they in people who take statins and people who don't take statins, yes, they are much, much more common in the people who take statins. But we already know that's going to be the case because there's other people with risk factors for which they take statins. So if you then put in conditions which can confound that association, so we put in diabetes, we put in everything like that, and then we look for an independent association, we see there's no independent association. So first we see, yes, we can see why people think this, because yes, in that statin group, more people do have cataracts. But we can also show that it's not independent, because if we put in our clinical conditions from those electronic healthcare records, we see that there's no longer an independent relationship between statin use and cataract. It's all down to the medical conditions for which you take statins. So that's the first bit of the analysis.
And then where does the protective effect of the gene get in? Because I think, like every biological problem, it's felt like you were just peeling back layers at the end. You seem to answer that first question really elegantly. Of most of the effect, or probably all of the effect, is driven by the fact that cardiovascular disease is correlated with or causal for cataracts, right? And so the statins are along for the ride, and you see that association because people are taking statins for those cardiovascular comorbidities, not because the statins are causing cataracts, which is all good. But then you notice an independent effect of some genetic subtype that was relating to cataracts independently. Maybe you can talk about that.
Exactly. So then we said, okay, if there's an independent association there, that's dose dependent, so something to do with, let's say, the pharmacokinetics, which means that, we look for a dose response curve. So we say people with more exposure to the medicine would more likely have the response that you think it's causing, right? So we say, in the group of people who receive statins, is there a difference in the rate of cataracts, young onset cataracts, in the people that have this genetic marker, which means they will have much, much higher exposure to statins, or the people who do not have the genetic marker, which means they will have lower exposure to statins. So we can say, okay, if they're actually causing cataracts, then surely getting more of that is going to cause more cataracts, right? And what we saw was the opposite, that people who have that genetic variant, where they're less able to clear out statins quickly and therefore they get exposure to higher levels of it, they actually had a 1/3 reduction in those young onset cataracts. So now we say, not only do we know statins are independently causing cataracts to happen, if actually you're getting a much higher dose by virtue of your genetic variant, you actually got a 1/3 reduction. You've got a protective effect from taking that statin. So that is quite useful to know, right? In terms of safety, that gives a lot of reassurance, and also maybe gives some mechanistic insight into some things we could use to help people to not have statins if they are in that high-risk group, because we know by definition people taking statins are in a high-risk group, right?
Yeah, that's super. I love that. And it's very detailed work. Like I'm sure that particular analysis took you months to put together, and that's the nature of research like this. It really takes time and care. And I guess what we're able to figure out whether okay, so I'm just going to repeat it because hopefully it's helpful for me and the listeners. The as you said very well right there, the presence of the genetic variant that you studied would mean that for some people they're getting higher, their body isn't clearing the statins as quickly or as effectively, so they're getting naturally higher levels. Those people have less cataracts. That could be mediated by the statins having some kind of independent protective effect against cataracts. Or I guess those people could be getting greater relief of their underlying cardiovascular symptoms, which then has the protective effect. Is it one of those two? Is it both of those? Is it hard to pick out? Does my question even make sense?
Yes, it's a fantastic question. And I'm afraid I'm going to give an unsatisfactory answer because the answer is that I would love to be able to answer that question. But what this tells us is sort of an indication of independent relationship between statin use and cataracts, and then between statin use and cataracts in people with that genetic variant. It unfortunately then can't offer the insight as to why that is. And I think there's another lesson here, which is quite important to discuss, which is also the other things it doesn't tell you, right? Because it's always equally important to talk about limitations and unanswered questions as well as something can answer. So we corrected for the presence of diabetes in this, right? Which makes sense because we want to know about independent association between medicine use and the cataracts. However, we know that statins have been associated with increased onset of diabetes, and we know that diabetes raises your risk of cataracts a lot. So for any question, for any design, you're able to provide some answers, but not others. And I think it's, important to stay honest about what you can't answer as well as what you can.
Yeah, that's so true. And you made a really important point, I think, in the discussion of the paper, which is that this effect, along with other genetic effects that you maybe weren't yet powered to discover or otherwise, could also explain some of the confusingness in the literature, right? If some studies find a positive direction of effect, others find a negative, then that could be because of a imbalance in the different genetic ancestries in the different arms of the study, right? Maybe you can expand on that a little bit because I thought that was a super point.
Oh, thanks. And I'm so glad that you wanted to speak about that because that's another thing that I think is really important to discuss broadly about pharmacogenetics. So we said before, right, that we're not doing a great job in science medicine generally of getting global diverse representation in therapeutics trials or in genetics, right? But we know that apart from sort of ancestral variation in genes, if you look at specific sort of subcohorts of ancestral groups in a national context, there also might be differences in terms of morbidity within that population, right? We might say certain groups maybe have higher rates of hypertension, diabetes, and that means that they'll have different prevalence of health events, right? So the issue with something like this, where we're talking about different prevalence of this gene variant across different ancestral groups, and also different prevalence of cardiometabolic disease, which is a reason you take statins, is that it can all get quite mixed up, right? So you could get quite different answers to that question you're asking, depending on which cohort you looked at in terms of comorbidities, deprivation, ancestry, and how that study was designed.
If you were to design clinical trials, for example, from a blank slate to try to solve for this problem a little bit better, would it be, and taking some of the operational challenges out of it, would having perfectly matched control and treatment arms down to genetic ancestry help to ensure these kind of effects didn't end up driving clinical trial results? For example, I'm wondering in some of these cases if there are trials that either succeed or fail by random chance because they haven't accounted for these kind of effects potentially.
It's a great point. So pharmacogenomics, I think, is now being used a lot within the drug discovery and development pipeline by industry, right? And that's twofold. So first, if you get the right subpopulation for medicine, then you can have a much more effective medicine. The other is de-risking medicine. So you can envision a scenario in which a medicine would be a really good medicine, but pose a risk to a small group of people that you could identify by genetics, right? But if you know that in advance and you're able to mitigate that, then you can save a really good medicine and protect the people who would be at risk from it. So there's a lot of potential there for de-risking as well. And I do think that industry is using that a lot now, and also coming through from sort of regulators and into clinical practice, we're going to start to see more of it. So the longest standing example of pharmacogenomics in practice is abacavir, a medicine for HIV. It's a very, very good medicine, but it used to pose a risk of a very bad sort of immune reaction to a small subset of people that could be identified by a genetic marker. So we figured this out as a scientific community, and therefore we saved a really good drug and we de-risked it. And now gold standard everywhere, before someone is offered that medicine, they get this genetic test. And if they have that risk allele for a bad reaction, they don't get the medicine. So that's a really good example. But we're seeing some much more current examples of medicines coming through the pipeline where pharmacogenomics will be incorporated to make sure the medicine is de-risked in the way it's given from the get-go.
I learned about an interesting example of this that's becoming more and more relevant, which is for some of the newly approved Alzheimer's drugs. There's a safety issue with people who are ApoE44 homozygotes have a much higher rate of, do you know about this, about the brain edemas? And I thought this was, this is a perfect example of exactly what you're describing, where people who carry the particular genetic subtype are at significant elevated risk of brain bleeding, right? And they've picked this up through clinical trials and post-market monitoring. But it begs the question of how you most effectively treat patients who carry these genetic variants who are going to face pretty significantly elevated safety risks, right? And this, we should assume this might be the case across every disease, right? Is that accurate? Rather than assuming it's not and being surprised.
It's a really good point. So this is an interesting class of medicines coming through for Alzheimer's. And the interesting thing about the risk of ARIA is that it's not just a safety signal. There's also the evidence showing that people with that ApoE4 genotype have less benefit from the medicine.
Right.
So it's not just higher risk, it's also a worse risk-benefit ratio on both sides, higher risk and lower potential benefit. So that's a particularly interesting example. There's also the new medicine, Magavern, which is a myosin inhibitor for patients that have HCM, a kind of inherited heart disease where the heart muscle is too thick. And this medicine, it was the first in class. Now there's other agents, but it is metabolized in the body, broken down by an enzyme encoded by a gene, which is very polymorphic and very well understood to be very polymorphic. And people who break it down less well because of very common changes in that gene get more of it, and therefore they're at higher risk of type A adverse drug reactions. Too much of a good thing, right? So if we say it does its job if it helps the heart relax, but if the heart relaxes too much, you get heart failure.
Yeah, this is a big issue. On the ARIA and the Alzheimer's one, there's a interesting company that I really like that is just focused right now on ApoE44 homozygotes. The company's called Alzion, and they have a small molecule. I think they're going to get their phase 3 readout halfway through this year. And I'm hopeful for that because it's a shame to have no good option if it sounds like there are safety issues, but also efficacy issues in this really important early onset population. So for those of you who are interested in that space, maybe a company or trial to watch out for. Do you think, with your medical doctor hat on, how should pharmacogenomics sit in healthcare? It is one of those things that we've talked about for a really long time, but seems stubbornly difficult to get healthcare systems to adopt. I think in part because the benefit is so diffuse, right? For one patient, it could be useful in the context of this discussion about about an Alzheimer's risk risk reward decision, and another one it could be for routine statin use. How do you think about that and solving the problem of making this broad use tool a little bit more available?
Yeah, it's such an important point, really. I know many of you will be aware of the recently published PREPARE trial results that came out in the Lancet, and that showed that sort of prospectively, if we look for drug gene pairs that we know have an impact on the risk of side effects and efficacy, we can decrease adverse drug reactions, unwanted side effects by 1/3 over a period of just 12 months. And we also know from different studies that adverse drug reactions are responsible for a high percentage of all hospital admissions nationally, 6.5%, and that the cost to the healthcare system is absolutely enormous. So recent estimates have placed it northward of 2 billion a year. So adverse drug reactions are really common and a really difficult problem that cause a lot of harm and risk to patients and a very high cost to the system. And I think the PREPARE trial convincingly showed that we can use PGX panels. It was just a 12 pharmacogene panel, so pretty limited, interacting with, I think, 50 or 60 medicines. And doing that, if we could decrease adverse drug reactions by 1/3, you can imagine the potential benefits at scale.
Yeah, and so where I'm not familiar with that study. I'm going to put a link in the show notes just in case other people aren't. But where I was just looking it up as you were talking. So it's a pan EU across I want to say 18 hospitals, exactly like you said, 12 gene panel. Where does something like that go from here? Do you think that the output of that trial is enough to get major trusts in the UK, for example, to adopt something like this, or is there still a missing piece?
It's difficult. I think with my clinician hat on, I would say that getting preventive care into the system is always a challenge because there's always a requirement for resources that are more reactive than preventive, right? You always have to solve the acute problem, and that makes sense because if you have to solve where to put the resources right now, of course you want to help the most acute needs first. But the fact is that preventive medicine is a lot of money up front, and then you see a lot of benefits, which I think have been really compellingly shown by people over the coming years. But certainly this is way above my pay grade, and I'm not qualified to say anything except sort of my own speculative opinion here. But I think that that it's really difficult to then get through things which have a much longer term payoff and a much higher upfront cost. So I think that the health economic studies coming through, which I know are pending also from the PREPARE trial, will be really useful because I think the arguments people need to see now are those health economic arguments. I think to put that money up front, the systems need to see absolutely indisputable evidence of long-term savings. So I think that's part of it. As a clinician hat on and a scientist hat on, I think that we're seeing worsening inequality and multimorbidity, polypharmacy, which are not proportionately distributed in our society. And this is troubling, of course, for many reasons, one of many troubling trends in inequalities, right? Um, and pharmacogenomics, therefore, if applied well, could potentially really address some of that for populations that have been underrepresented. There's also a real risk here, though, which is that if people don't work with communities that are already underrepresented and have already sort of been less present in trial evidence and sort of the evidence from which we extrapolate our knowledge, then actually they could make things worse. And that absolutely must not happen. So I think that communication, engagement, and really working with all the communities who have a lot to gain from having more evidence base in these therapeutics and de-risking and making sort of more efficacious these therapeutics is of the utmost importance, if that makes any sense.
Yeah, absolutely. And I think it dovetails into one of the other I think it was maybe a release around this paper where you were quoted just talking about the importance of building trust with the community and not taking that piece for granted because we know that our health data is one of the most sensitive things that we have, right? And if you lose your access to your social security number in the US or the UK equivalent, that these are things that unlike a bank account, you can't call them up and say, I've been, I've lost my card, please shut it off. If you've lost it, you've lost it, right? And so there's a importance for people to maintain that trust and control, but there's also the backdrop of a lot of historical wrongs, right, that we need to think about when we're running research. So maybe you can talk a little bit about the community engagement side of this work. It's one thing to come up with a plan for your research, but then how do you actually work with genes and health in this case to put that into action?
Oh, quite right, quite right. So Genes and Health has done an incredible job of making the study possible and working with the community. Have I lost you? Oh, I've just still got you. Sorry, my computer
Yes.
just being funny. And working with the community. But they've got a very good community advisory board, and they have a whole recruitment team that have strong links within the community, and they've had strong endorsement from community members. But you're right, of course, the history of mainstream establishment with groups that have been underrepresented in studies and interfaced less with sort of medicine, academia in terms of clinical trials and research. There's a lot of reason for that, and there is mistrust, which is very well founded from a long history of trust not always being earned. So I think that it's a really big issue, and one which I, again, am not qualified to give an appropriately big answer to. So what I personally feel is the best that I can do on sort of my level is communicate and to try to advocate for inclusion of diverse perspectives and diverse data. So I did some sort of focus group works with the participants in the genes and health community around pharmacogenomics. And I'm doing some more now on broad sort of public acceptability of both clinical implementation of pharmacogenomics, but also broader sort of research from potential clinical data to find out from the get-go, what do people like, what do they not like, and what are they worried about? Because I feel that if you ask first what
Yeah.
concerns are and what safeguards we could put in place, that then there can be maybe a more constructive dialogue, because then you can put in place the measures, the safeguards that maybe you wouldn't have thought about, but they are what people are worried about. And I think that's really important, to make that something that comes from the community and then feeds into policy, so that you're making that research safe and comfortable and de-risking it.
Yeah, absolutely. So, as we wrap up here, what's next for you? What are the one or two projects that you're most focused on that you can talk about? I know sometimes things are too early to want to speculate on any results, but a general direction of travel and what's next would be really interesting.
So I think that there's sort of three streams here. One, my academic research. I think there's so many infinite questions that we can ask with sort of big scale data on pharmacogenetics. So using genetic tools to understand better sort of sub-endotypes of disease. So what we might be grouping together that actually are a bunch of sort of slightly different diseases. And then also differences in response to treatment. Because by understanding better what exactly the cause of the problem is and why exactly some people respond better or worse to a medicine, we can do a much better job at treating it, right? So one stream is that research avenue. Another is sort of policy-oriented, public acceptability of pharmacogenomics. So essentially talking to people, but a slightly more academic tilt a bit. So doing more public engagement, focus groups, surveys, with big stakeholder organizations within the UK, so that we can understand from people how they would want pharmacogenomics to work for them in clinic,
Yeah.
so that then we can try to make that possible. And the third stream is trying to bring into clinic as much as we can within the UK, with all of the colleagues working on this wonderful stuff, the gains of pharmacogenomics sort of from the bench to the bedside when appropriate.
What do GPs, is it GPs who'd be the primary recipients of this? What do they think about pharmacogenomics? Obviously there's a huge spectrum, but I'm curious of some of the themes that you're hearing.
So there are some sort of champions of pharmacogenomics within the GP community in the UK that have done a lot of wonderful work around pharmacogenomics. And I had the great privilege to work with some of them on a Royal College of Physician report. So it was Jude Hayward and Imran Rafi worked on that with us, and they've been great champions of pharmacogenomics within the primary care space in the UK. I would not speak for them. They've got all that expertise. But I think, there's interest, but GPs are very busy and they've got a lot on their plate. And I think the consensus is that really there's got to be a very smooth system in place with very good clinical decision support, because GPs are already overburdened and this needs to be something that makes life easier and not harder and is usable. So it needs to come with a whole system of not here is someone's genotype, but this is what you should do with their medicine.
Yeah, that makes total sense. Well, this was super fun. I appreciate you walking me through the great paper that you wrote and also some of the work that you're doing besides this. If people want to keep track of you and your work, what's the best place to find you? Would it be on the Queen Mary website? Do you use Twitter, or have you stayed away from that dumpster fire? What's your social media?
I'm going to guiltily confess to being an absolute Luddite. It's terrible of me being a data scientist, but I actually never joined any form of social media. And I'm quite happy with that decision.
I think you've done fine there. You don't need to make a Twitter account today.
Apologies for that. But yes, certainly via Queen Mary, and always delighted to speak with people about research and sort of any discussion at all is always good discussion.
Great. Well, thank you. And thanks everybody, as always, for listening. If you enjoyed this episode, the thing that I ask you every episode that I really appreciate is just share it with a friend or a colleague who you think would like it. And of course, you can always leave us a review on your favorite podcast player. Thanks again, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.